Welcome to this video demonstrating the HIT Confirm test. The video aims to present the test step by step and to highlight the important points. Make sure to familiarize yourself with the instructions for use, checklist, and frequently asked questions document before watching this video. Preparing patient plasma. Obtain fresh plasma from the patient's citrated whole blood. The test can be performed directly from this plasma or from aliquots stored at under -20 degrees. In this case, quickly thaw the sample to 37 degrees. The plasma must be filtered if stored frozen. Use filters with a membrane pore size of 0.2 microns. Spin filters are preferred for their lack of dead volumes. Keep the plasma between 20 and 25 degrees throughout the entire test. Before each use, homogenize the plasma with a vortex mixer. Preparing platelet-rich plasma from a healthy donor. Obtain citrated whole blood from healthy donors, preferably from an authorized center. Once received, gently homogenize the tubes, then let them sit still for at least 30 minutes. Homogenize the tubes again. Prepare the PRP by centrifuging between 20 and 25 degrees at 200 G for 5 minutes without using the centrifuge brake. In this video, the test is performed with 2 PRPs prepared from 2 different donors. Collect as much supernatant-containing platelets as possible without perturbing the 2 phases and transfer it to a new tube. Verify that this supernatant is cloudy, indicating enough platelets are present. Avoid forming air bubbles. It is essential to observe the whole blood and PRP shelf lives indicated in the instructions for use. Performing the test. Once the plasma and PRP are ready, quickly proceed with the test. All the steps are performed between 20 and 25 degrees. One kit contains 6 reagents, 1 dilution buffer, 1 orange tube of anti-CD41 antibodies, 1 green tube of anti-CD62 antibodies, 1 yellow tube of heparin at the concentration of 3 units per milliliter, 1 blue tube of heparin at the concentration of 1,000 units per milliliter, and 1 tube of trap. Before each use, homogenize and centrifuge all tubes contained in the kit. When first using the kit, reconstitute the trap at 50 microliter of dilution buffer. Four tubes need to be prepared for each test: neg, pos, HO.3, and H100. These 4 tubes contain the same volumes of dilution buffer, the 2 antibodies, and PRP. For simplicity and reliability, prepare a master mix. The mix contains 115 micrometer of dilution buffer, 5 microliter of anti-CD41, 5 microliter of anti-CD62, and 50 microliter of gently homogenized PRP. Gently homogenize the mix and put 35 milliliter into each tube. Add 5 microliter of 3 units per milliliter and 1,000 units per milliliter of heparin to the HO.3 and H100 tubes respectively. Then complete with 50 microliter and 10 microliter of plasma. Complete the 50 microliter neg and pos control tubes with 15 microliter of dilution buffer. Lastly, add 2 microliter of trap to the pos tube. Incubate all 4 tubes between 20 and 25 degrees for 30 minutes in the dark. Add 450 microliter of dilution buffer to all the tubes and gently homogenize. Load the tubes into the cytometer and immediately start the reading in the following order: neg, pos, HO.3, and H100 according to the previously established model using Appendix 1 in the instructions for use. This model contains settings for the FSC threshold, voltages and gains, and compensations, as well as 3 graphs: a log FSC log SSC dot plot, a PE histogram, and a FITC histogram. Read the neg tube by collecting 10,000 events in the platelets window at slow flow rate. Adjust the FSC threshold and the platelets window if needed and restart the acquisition. Obtain the following profiles for all 4 tubes. Analyzing and interpreting the results. The log FSC log SSC density dot plot represents the 10,000 collected events. These events should correspond to the platelets present in the PRP and must be displayed as a single population in all 4 tubes. Superimpose the PE curves obtained with all 4 tubes. Verify the presence of a single platelet population with similar profiles in all 4 tubes. Make sure that at least 95% of the events are present in the platelets window for the neg and pos tubes. Activate the platelets window on the FITC histogram. Superimpose the curves for the neg and pos tubes and place a vertical marker at the intersection. This marker defines the activated window. Read the percentage of events within the activated window and make sure that neg and pos values are in the acceptable ranges specified in the instructions for use. Under the same conditions, superimpose the curves for the neg and H100 tubes and make sure that their profile is similar. Lastly, superimpose the curves for the HO.3 and H100 tubes. Read the percentage of events within the activated window. Calculate the HEPLA index using the following formula. If a result lower than -13% or between 9.6% and 13% is obtained, the test should then be repeated with a different PRP. If any of the instructions described in this video is not followed, we recommend repeating the test with PRP from a different donor. Thank you for watching this video. Do not hesitate to contact Emosis or one of its sales representatives for any additional information.